Hello everyone, welcome to Big Orange Biology. Today we're going to talk about cell organelles and their structures. Now, to really get into cell organelle structure and function, we need to really just go ahead and formally talk about all the various organelles there are in the cell. And for this video, we're going to start from the nucleus out. But also we're going to talk about the reason why cells need organelles. This video, I will be giving you a heads up, we'll move at a fairly brisk pace. For that, I will be sure to include the power plant with this one below if you wish to look at that. I will elaborate on it as needed. This is mainly for those who wish to listen to the video. That said, class is in session. Let's begin. Okay, so let's talk about why cells need to have organelles. So most cells generally are tiny. They do this in order to maintain what we call a surface to volume ratio. The exact surface volume ratio depends on the cell and its unique function, but it mainly comes down to the fact that as a cell gets larger, it has to worry about metabolic activity. The bigger the cell, the more metabolic activity it needs. And that means it needs more resources. Well, surface area is what's going to determine how many resources can go into the cell and how many waste products can exit the cell. So typically, the less surface area you have to your volume, the more inefficient your cell is going to become. Now, to mitigate this, cells do adapt some various means. This here is a graphical representation explaining why surface area to volume is kind of difficult. See, surface area is a squared function, while volume is a cubed function. Hence the cubes. Silly joke. But as you increase the size of a cube, let's say we start with a 1 millimeter cube, so it's got 6 millimeter surface area, and its volume is 1 millimeter cubed, obviously. But if you, say, increase the size to 2 millimeter cube, you get a 24 millimeter squared surface area, cool, but an 8 millimeter cubed volume. That results into a surface area volume ratio going from 6 to 1 to 3 to 1. Increasing by 1 causes the surface area to volume ratio to decrease by half. As we see here, it goes from 6 to 3, then 3 to 1.5, and so on and so forth. So certain cells need to mitigate this issue. So the general rule is as cells get larger, metabolic area and the need for resources can tend to increase faster than the surface area can keep up. So bigger cells, more volume, have a hard time keeping up with their surface area. So to accommodate for this, many large cells will actually increase the surface area by doing a lot of things to the membrane, increasing folds, making the cell a lot more narrower in places. There's a lot of adaptations we will explore in AP Bio as the course goes along. Now, the cell membrane is the outside of the cell, so we're going to kind of talk briefly about this. Then we're going to go inside out. So a cell membrane plays a key role for the cell. The cell membrane's main role as a selectively permeable membrane to help maintain homeostasis. This homeostasis process includes things like water and salt levels and various other ions. Also, the cell membrane's main function, in addition to being a semi-permeable membrane, is involved with cell signaling and communication, and also helping hold on to other nearby cells to kind of function as tissues. Now, before we really get into all the organelles, we need to differentiate between the two types of cells. Just a reminder, there's prokaryotes and eukaryotes. Prokaryotes are your non-membrane bound organelles cells. These are the most simplest cells we typically see. They have a DNA located in what we call a nucleoid region. That means there is no nucleus, and there's no other membrane-bound organelles like mitochondria and vacuoles. Contrast that with the eukaryote. Now, the eukaryote is your membrane-bound organelle system, including the nucleus. They're notable for having the nucleus. That's actually the definition of a eukaryote, contains a nucleus, a true nucleus. And they also have other membrane-bound organelles. Now, eukaryotic cells are also optimized by using these organelles, like I mentioned. Organelles help increase the efficiency of the cell. By having these tiny little specialized compartments, it allows the cell to be more efficient and carry out its various cellular functions, allowing for the cell to grow increasingly larger in the process. So not only does volume to surface area matter, it also matters how you utilize that volume. And eukaryotic cells find a more efficient way by specializing certain regions of the cell to carry out specific functions. These specializations allow the cells themselves to become more specialized when time comes for them to interact with and develop into other cells. So each of these all do play a key role here. So we're going to start with one structure that all life has, regardless if it's a prokaryote or eukaryote, and that is the ribosome. Now, the ribosome is a key enzymatic process. It contains RNA and proteins. The ribosome's main job is to make more polypeptides. It reads mRNA and translates it into the appropriate amino acid polypeptide sequence. Now, the nucleus is typically the largest organelle. Now we're looking at the eukaryotic category. So we're going to start in the center of the cell. So we're looking at the eukaryotes here, and the nucleus is the largest one of the organelles, typically. It's the location of DNA and DNA synthesis. It's also where DNA is going to be transcribed to RNA using what's called transcription. It also contains a smaller structure known as the nucleolus, which is the main site of ribosomal and protein synthesis. Now, the nucleus itself is surrounded by two membranes that function much similar to the cell membrane. The nuclear pores on the membrane allow certain things to go in and out. It allows the membrane to be, controlled as a semi-permeable membrane, and that allows for the DNA to be extra protected. It also allows the nuclear pores also allow the RNA sequences to leave and other chemical signals to enter. Now, in the nucleus, DNA can also be found in a form of what's called chromatin, these long, thin strands, and it's usually found in the more common form known as chromosomes. Most of us know what a chromosome is. Doubtfully, you know what a chromatin is. Chromatin is just DNA with proteins here, and when you condense them, you get yourself a chromosome. Now, the outer membrane of the nucleus is going to be attached to the endoplasmic reticulum. So the outer level of the nuclear membrane is attached to the endoplasmic reticulum, taking us to the next site, the endomembrane system, which includes the endoplasmic reticulum, nuclear envelope, Golgi apparatuses, and the lysosomes. Now, also there's going to be these tiny little membrane-bound organelles known as the vesicles to help shuttle substances throughout the cell, mainly proteins and lipids and other chemical signals that may need to be inside the cell and possibly to the outside of the cell. Now, the endoplasmic reticulum is probably the notable member of this group. This is the network of interconnected membranes in the cytoplasm with large surface areas. There's two types of endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum. Now, rough endoplasmic reticulum has ribosomes attached to its outermost surface. These are where newly made proteins are going to be made. They're going to be made in what we call the lumen region. It's a special region within the endoplasmic reticulum. Don't get too hung up on that. But that's where the proteins are going to be going from the ribosomes inwards, and when they get in there, they're going to be chemically modified and tagged for special delivery. This is where tertiary structures and quaternary structures also take place. The endoplasmic reticulum helps facilitate this. Now, the proteins are then going to be transported by vesicles to other areas, and these vesicles will be pinched off of the endoplasmic reticulum. All secreted proteins and most membrane proteins will pass through the rough ER. Now, polypeptides that are transported in there will make the tertiary compounds, as I mentioned. Some will also be linked to carbohydrate groups, becoming what we call a glycoprotein. Glyco meaning sugar. So it's being attached to a sugar. These are very important for interaction and recognitions between cells. They're going to go straight to the cellular membrane for cell signaling and identification purposes. Now, the smooth ER is not to be left out. Most people tend to neglect that. Smooth ER plays a very critical role. While it is tubular and no ribosomes, it is the organelle that chemically modifies small molecules such as drugs and pesticides. That means it is the purifying region of the cell. Typically, a lot of chemicals like alcohols are going to be detoxified here. It's also the site where glycogen will be degraded in animal cells to break it down for smaller purposes. It's also the site of protein and lipid synthesis. Sorry, not protein, steroid. Lipids and steroids are going to be synthesized here in the smooth endoplasmic reticulum. It's also where calcium ions are going to be stored for proper cell responses, which we will discuss in later units. Now, the Golgi apparatus is the flattened sacs that are found within this endomembrane system. These receive proteins from the rough ER and even further them. So it receives proteins from the rough ER and further modifies them to make them a little bit more compact. So we mess with that tertiary and quaternary structure even more. It helps concentrate, package, and sort the proteins and adds carbohydrates to making more glycoproteins. And it's also where polysaccharides such as cellulose are going to be made for plant cell walls. Now, we're going on to the lysosomes. So lysosomes are coming from the Golgi apparatus, so we're going to get the primary lysosome. The primary lysosome contains the enzymes known as hydrolases. These are the ones that do hydrolysis, which we discussed in previous unit. This is the process of breaking down macromolecules. So this is where the hydrolyzing molecules needed to break down, like proteins, lipids, carbs, and nucleic acids are going to be found. Now, primary lysosomes are going to basically be involved here, because what's going to happen is, as a macromolecule enters the cell by what's called phagocytosis, which then the cell membrane wraps around a solid substances and brings it in, making a phagosome. The phagosome will fuse with that primary lysosome to form a secondary lysosome. And the secondary lysosome will hydrolyze the food molecules into the proper monomers that are needed for the cell to function. Now, phagocytes are cells that help specialize breaking down and taking in materials such as carbs, lipids, proteins, and nucleic acids. Now, autophagy is the process which lysosomes will break down cell components. So some lysosomes are known as a cleanup. That's probably what you learned it as in middle school. So when the lysosomes come around, they're going to break down the other cells, materials, and even entire organelles that are outdated and outworn out. And they're going to break them down to their subparts and transport those parts elsewhere. Lysosomal diseases, by the way, because if lysosomes fail to clean up, are going to be when they're going to create various disorders, possibly linked to even diseases like Parkinson's, where lysosomal disorders, basically the cell can't clean itself up, and that causes various other errors to occur. Now, in eukaryotes, breakdown of energy-rich molecules will begin in the cytosol. That process is called glycolysis. This is also true for prokaryotes. But after that, they'll go to where it's called the mitochondria, commonly known as the powerhouse of the cell. Please never call it the powerhouse of the cell in my course. Mitochondria is where energy-rich molecules are going to be converted to ATP. So we're going to take in carbs, proteins, nucleic acids, and lipids. Carbs, proteins, and lipids. And we're going to break them down into energy-rich ATP. We typically look at cellular respiration, which is the breakdown of carbohydrates. Now, cells require a lot more energy, typically have mitochondria, so things like neurons, muscle cells, etc., and those like will have more mitochondria compared to cells like your skin cells, which may not need to do a lot of movement. Now, the mitochondria is very unique, and we'll explore its structure and function in greater detail in the third unit. However, we do need to talk about its unique thing about structure, because its structure indicates that it has pulled in another cell at some point in time. It is a endosymbiosis example. Mitochondria have two membranes, an outer membrane, which is very porous, allows everything to go through, and an inner membrane, which has got extensive folding, much like various prokaryotes, that allows for increased folding called cristae. That allows more surface area. More surface area means more place for reactions to occur. The fluid-filled matrix will contain enzymes such as DNA and ribosomes. Now, DNA and ribosomes are going to be in there, so that means it has very much the similar characteristics of a prokaryotic cell. It just cannot live on its own, so now it's called an organelle. Now, mitochondria has a similar organelle out there called the chloroplast, and a chloroplast is a part of a group of organelle structures called plastids that are found in plants and algae. These are plastids are things that can differentiate further into various organelles. The chloroplast is the most famous. It contains the pigment known as chlorophyll, and it's where photosynthesis occurs. It is also where it's going to convert light energy into chemical energy. Now, chloroplast, much like the mitochondria, has two membranes, and the inner membrane is called the thylakoid. Now, the thylakoid is differentiated into what's called grana, which are a stack of various thylakoids, and where the light energy reactions take place. Now, the stroma is the aqueous matrix that are found around the grana, and that's where carbohydrates are going to be synthesized. Further on this in Unit 3, which is all about energetics. Now, other plastids are things like chromoplasts, which make plants look pretty by giving them various colors, and other specialized organelles now. Now, other organelles that are key to the cells are peroxisomes and galaxosomes. Peroxisomes collect and break down toxic byproducts of metabolism, such as hydrogen peroxide, and they use specialized enzymes to do that, known as peroxidase, for instance. But other enzymes are going to be involved here for other chemicals. Galaxosomes help break down lipids, and they're going to convert them into carbohydrates that plants need for growth. Now, let's say what you do with all this material as you collect it. So you have breaking down materials. You have lots of breaking things down. You have ATP being made in the mitochondria. And you have carbohydrates being made in photosynthesis all over the place. What are you going to do? You need to store them somewhere. So vacuoles come in here. Now, vacuoles are the main storage molecule of the cell. This is where we're going to have different functions. It's where various waste products are going to be stored. This may actually be where you can store a bunch of toxic chemicals to help deter herbivores. And in plants, it also plays a key role in what we call turgor pressure, which we'll talk about further in this unit. Basically, you want to get a lot of water in a plant cell, and it's where you keep a lot of that water. Now, the cytoskeleton is the thing that helps maintain and support cell shape. Holds organelles in position and helps move organelles around. The cytoskeleton plays a very key role, and it's one of the defining characteristics of a eukaryotic cell. It is also going to be used in things like mitosis. It also has extracellular structures to help keep cells in place. Now, extracellular structures, so we're now moving to the outside. So we've now gone past the cell membrane and we're outside of the cell. And there's two different types. In eukaryotes, these structures are typically macromolecules and gel-like mediums. In plants, they also have a cell wall, which is a semi-rigid structure that helps provide support for the cell. As seen here, remember, it is located on the outside of the cell membrane, not the inside of the cell membrane. Plant cell wall has three major functions. It provides support for the cell and limits its volume by remaining rigid. It acts as a barrier for various infections, and it helps contributes to plant growth during development. Now, adjacent plant cells are going to be connected by channels called plasmodesmata, and these are going to allow certain things like ions, small molecules, hormones, and even RNA and proteins to transfer across, allowing for signaling to occur. Now, animals are not alone in this. Animals do have extracellular matrix. It's called made out of collagens and proteoglycans, and they help hold them all together with a third group of proteins, as we see here. Now, extracellular matrix is great because it holds cells together in tissues and also contributes to things like cartilage, skin, bone, and other tissues. It's used for filtering materials, think your kidneys, and helps orient cell movement during tissue repair. Lastly, now let's say we have a bunch of cells and they're touching each other. How do they touch each other? Well, you get junctions. Junctions are these structures that protrude from adjacent cells and kind of glue them together. Tight junctions hold everything together and basically makes a watertight area that only allows certain things to travel through. And it makes it much more restricted. It's great for epithelial cells because you want certain things to stay in or stay out. Lysosomes are going to link adjacent things and allow some things to travel through, and things like your plasmodesmata stuff. And gap junctions are going to allow for greater transport of materials, greater molecules to pass through here. Now, in conclusion, we're done talking about all this here. This was a rather quick video. I know it went at you very fast, but what you need to understand that cells have all these various organelles to help carry out their complex function. Eukaryotes are very large and they need a lot of different compartments to help carry out specialized functions so they can remain efficient. Now, we're going to explore each of these further in class and also in your disease research project that has been assigned to you. But if you have any questions, don't hesitate to ask. This was meant to be a brief, albeit very quick overview of the cell organelles. Just remember, the cell organelles help increase the overall efficiency of the cell, and certain cells will have certain organelles more so than others. Thank you and have a great day.